Good evening, students. Madam Winnie here. Biochemistry Simplified. It has been long since I upload a bit of the contents, but I'm back. I've been a bit busy, but I'm back. I would like to talk much about the third law of Mendel, the independent assortment. I already discussed the other two, the law of dominance and the law of segregation. So today, let me wrap it up by looking at the third law of Mendel, which is the law of independent assortment. What does the law say? It says that the members of one gene pair, we are talking about the alternate forms of a gene, that is the alleles. So the members of one gene pair will segregate independently from other gene pairs during gamete formation. So in this case, we are talking about gene pair. We are talking about a case where we have alleles, which are the alternate forms of that gene, a gene controlling a trait, and then the other gene controlling another trait. So we are talking about a dihybrid cross. In the case where, for example, we have the gene which controls shape, it's either round or wrinkled. So the possible alleles, the alternate forms of the gene, will have the dominant one R for round and the small r for the recessive, which will represent the wrinkled. Then you have another gene, possibly height. So the gene will have the alleles, the alternate forms of the gene will have capital T for tallness, small t for short. So we have two genes, but they are saying the members of one gene pair, as much as these genes are controlling two traits, they have to be independent of one another at the point of gamete formation. So they talk about they don't depend on another. If it is the gene which controls shape, it independently segregates from the gene which controls height. So they talk about alleles for different traits. Alleles for shape, we have the capital R, dominant for round, small r for wrinkled. That is allele for the gene which controls shape. Then we have alleles for the gene which controls height. I've given, for example, capital T for tallness, small t, which is recessive for shortness. So we talk about these two genes which are controlling those traits. They are distributed to sex cells, that is the offsprings, the F1s, if you have the parental generations, independently of one another. And this law is expressed well using the dihybrid cross. So when I talk about dihybrid, we are dealing with two traits, and that's why I gave example of shape and height. Talks much about the dihybrid cross being an example of the type of cross which expresses much of independent assortment as the third law of Mendel, or the third principle of Mendel. So it tracks the inheritance of those two traits. The two traits, we're talking about two genes controlling them. Each of the gene has two alleles. We're talking about a biallelic system. The alternate forms of those genes, they have the contrasting trait, so they will control those two traits, and those genes, they will independently segregate. So that is what is known as the law of independent assortment. But then, as much as each of these alleles segregate independently at the time of gamete formation, we have to see how we can generate the gametes. In the case where you have the two genes which control the two traits, we have to generate the gametes. And in generation of gametes, we are talking about identifying the number of heterozygous. And the formula for identifying the number of heterozygous, so that you can proceed and see the number of gametes which are generated for any of those cross which you have, then definitely we have the formula 2 power n, where n represents the number of heterozygous. How we can generate the gametes, or how you can talk much about the number of gametes in the population you're working on. Still, we are stressing much on the dihybrid cross, explaining much about the independent assortment. So as much as we can remember that the formula is 2 to power n, where n represents the number of heterozygous, the example we have 3 of them, we can see, you can point out the number of heterozygous in this state. So if I have to talk about the number of heterozygous, then definitely I will highlight here. We have 1 and 2. So we have 2 of heterozygous. It means we are going to generate gametes 2 to power n. N is 2. So we are going to generate here 4 gametes. In this case, we have how many heterozygous? 1, 2, 3. 2 to power 3, meaning 2 times 2, 4 times 2, 8 gametes will be generated. Then example here. So the generation of the gametes, that is what will lead us to understand much of the independent assortment. 3. This was homozygous, homozygous, heterozygous, homozygous, heterozygous, heterozygous. So we have 1, 2, 3, 4, 5, 6. 2 to power 6. This one is 2 to power 3. This one is 2 to power 4. So for us to know the number of gametes, then we need to know the number of heterozygous, our sequence. So in this case, we are talking about 2 to power 2. We have 4 gametes. The second one, we have 8 gametes, and then the last one, we have 64 gametes. So we have to be guided by the number of heterozygous. And as we have mentioned, for us to understand the independent assortment, we'll be able to highlight the point that the dihybrid cross will help us to understand much how the independent assortment as a law is understood when Mendel came up with this law. So what we are going to do, for example, here we have 2 traits, seed shape and seed color. In this case, you have to mention the traits up there. If you see such a question, very simple. You have to mention the law, and secondly, you have to generate the gametes. But before you generate the gametes, you are having 2 traits, so you have to clearly show the traits up there and the alleles for each of the genes controlling the trait. So for the trait which we are talking about, seed shape, it's either round or wrinkled, but all along round has been dominant over wrinkled. If you use one letter, the capital one, as we mentioned by the time we are introducing genetics, is that the capital one will have to represent the dominant allele, and then the recessive allele will be represented by the lowercase of the same letter. And then to the another trait, which is seed color, capital Y is dominant over small y. So capital Y will represent the yellow color. It is dominant over green. Then we said that we are going to generate 4 gametes. So what you can see here is we have the dominant allele for round, recessive allele, wrinkled. This is the gene which controls shape. Then we have capital Y for yellow, small y for green. This is the gene which controls color. So for us to generate gametes, what you do is just cross capital R with capital Y. You'll get capital R, capital Y. Still with the capital R goes with small y. You'll have capital R, small y. Small r, capital Y. You'll have small r, capital Y. Then small r, small y. You'll get small r, small y. The same will go for, remember, the male and the female. They have to contribute equally. So at this point, you'll have the male and the female, also the same number of gametes. So you have all the possible gamete combinations as capital R, small y, capital R, capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. The same with the male and the female. So we are talking about they independently segregate at the point of gamete formation. So you proceed with the same. We promised that the Punnett square is going to help us to solve all the genetics problem. So here you put the male is always on the head, it's the head one. So you put it up here, and then the female will come here. They equally will contribute. Then as you do matrix, in mathematics, you go with the first row, first column. You'll have capital R, capital R, capital Y, capital Y, until you fill all the boxes. So you will have the F1s as this. So capital R, capital R, capital Y, capital Y. But then generally what it will tell us much of the phenotype is when you have the capital R, capital Y, it talks about round and yellow. So it means even in the case where we have capital R, small r, capital Y, small y, it will still be round and yellow because the law of dominance will apply. Only where you have small y, capital R, it will be round green. Then we will have wrinkled yellow. It means you will have small r, capital Y, or capital Y, small y. Then they will be wrinkled yellow. In case where you have wrinkled green, you will be having small r, small y, small y. So the phenotypes will be spread in the ratio of 9 round yellow, 3 round green, 3 wrinkled yellow, and 1 wrinkled green. So you'll talk about the phenotypes. Then when you're told about the phenotypic ratio, you have to give 9 round yellow, 3 round green, 3 wrinkled yellow, and 1 wrinkled